Hello, this is Dr. Morris continuing with the lecture series, Chapter 2, Part B, Chemical Principles. Important biological molecules, organic compounds always contain carbon and hydrogen. Inorganic compounds typically lack carbon. Inorganic compounds such as water are essential for living cells. Water, which is a polar molecule, has an unequal charge distribution that gives it four important characteristics, and you should be familiar with these characteristics. First, a high boiling point that, incidentally, a solid such as ice that is less dense than liquid, a characteristic that allows it to float. Number two, excellent solvent properties, that is, as good as dissolving medium. Polar substances undergo what's called dissociation into individual molecules in water. They dissolve because the negative part of water molecule is attracted to the positive part of the molecule in the solute or dissolving substance. 3. An important role as a reactant or product in many chemical reactions. And 4. An excellent temperature buffer capability that helps protect the cell from changes in environmental temperatures. This is the structure of the water molecule, a polar molecule. This is an illustration of the polar substances that dissociate, forming solutes. The hydrogen atom and hydroxide atom participate in chemical reactions. Hydrogen bonding between water molecules makes water a temperature buffer. When inorganic salts, such as sodium chloride, are dissolved in water, they dissociate and undergo what's referred to as ionization. An acid dissociates into one or more hydrogen ions or protons, and one or more negatively charged ions. In other words, acid protons, donors, in HCl, for example, yield H plus Cl. A base such as sodium hydroxide dissociates into sodium and the hydroxide ion. A salt is a substance that dissociates into ions that are neither hydrogen or hydroxide in nature. Sodium chloride is an example. It dissociates into sodium ion and the chloride ion. An acid is a substance that dissociates into more hydrogen ions. HCl yields hydrogen ion plus chloride ion. A base, a substance that dissociates into one or more hydroxide ions. Sodium hydroxide, for example, yields sodium and the hydroxide ion. A salt is a substance that dissociates into cations and anions, neither which is hydrogen nor hydroxide in nature. Sodium chloride, again, sodium and the chloride ion. The pH of a solution is the negative logarithm to the base 10 of a hydrogen ion concentration in moles per liter. Acidic solutions contain more hydrogen ions than hydroxide ions and have a pH lower than 7. That's an important concept that you should be familiar with. Alkaline or basic solutions have more hydroxide ions than hydrogen ions. A pH of 7 with equal concentrations of hydrogen ion and hydroxide ion is considered to be neutral. Each change in the number of logarithmic pH scale represents a tenfold change in the concentration. Buffers are compounds that keep the pH from changing drastically. The amount of hydrogen ion in a solution is expressed as pH. pH is the negative logarithm of the hydrogen ion concentration. Increasing hydrogen ion concentration thus increases acidity. Increasing hydroxide ion concentration increases alkalinity. Most organisms are best between the pH of 6.5 to 8.5. In humans, the ideal pH is 7.35 to 7.45. You should be familiar with this entire slide. All of the information on it is testable material. This is the acid-base balance. Gives you some idea of the substances both in the environment and those substances inside the human body and what their typical pH is. I make note of stomach acid, which is around a pH of 1. Note that the pH of 1 is very close to the pH of hydrochloric acid. As you can see, as you go to a more increasing basic solution or alkaline solution, you can see oven cleaner has a pH that's around 13.5. Pure water, milk, around a pH of 7, which is neutral. pH of urine is around 6. All organic compounds contain carbon, whose 4 covalent bonds allow it to combine with neighboring or other atoms to form large structures. Hydrogen fills most of the free bonds in these structures. There are many other functional groups, which as the hydroxyl group OH, not to be confused with the hydroxide ion OH of the bases. Such fundamental groups can be added to the carbon skeleton to make alcohols or amino acids. When single bonds hold a carbon atom to 4 other atoms to form the skeleton three-dimensional shape, it is called a tetrahedron. Such organic molecules can be combined into very large molecules called macromolecules. Generally, these are polymers which are formed by repeating small molecules called monomers. When 2 monomers join together, they usually release a molecule of water, a reaction called a condensation reaction or dehydration synthesis. The chain of carbon atoms in an organic molecule is referred to as the carbon skeleton. Functional groups are responsible for most of the chemical properties of a particular carbon compound. These are the representative functional groups that are typically involved in the human body: the alcohol group, the aldehyde group, the ketone group, methyl group, amino group, and the ester group. Ether groups, sulfhydryl group, carboxyl group, and then the phosphate group. You should be somewhat familiar with these functional groups and their biological importance. This table is taken from your textbook. Small organic molecules can combine in large macromolecules. Macromolecules are polymers consisting of many small repeating molecules. The smaller molecules are called monomers. Monomers joined by dehydration synthesis are condensation reactions, as seen below. Carbohydrates, important part of the cell structure in the human body. Carbohydrates have a general formula of Ch20 with a hydrogen-oxygen ratio of 2:1. Carbohydrates form monosaccharides, simple sugars, 3 to 7 carbon atoms, disaccharides, 2 monosaccharides bonded together. Sucrose, for example, is made up of glucose and fructose. And polysaccharides, 8 or more monosaccharides, such as glucose joined by dehydration. Familiar examples are cellulose, starch, and chitin. The latter is found in the cell walls of fungi, the exoskeleton of lobsters, crabs, and some insects. Chitin. 2 molecules with the same chemical formula but much different structures are called isomers. An important structure and as energy sources consist of carbon, hydrogen, oxygen with the formula Ch02o. Below, again, an example, you can see commonly the dehydration synthesis and hydrolysis that's involved in this process. Monosaccharides are simple sugars with 3 to 7 carbon atoms. Disaccharides are formed when 2 monosaccharides are joined in a dehydration synthesis. Disaccharides can be broken down by hydrolysis. Oligosaccharides consist of 2 to 20 monosaccharides. Polysaccharides consist of 10 or hundreds of monosaccharides joined through dehydration synthesis. Starch, glycogen, dextran, cellulose are polymers of glucose that are covalently bonded differently. Chitin is a polymer of 2 sugars repeating many times. Lipids, another important constituent in the cell membrane, are the primary components of the cell membrane. Consist of carbon, hydrogen, and oxygen. They are nonpolar and insoluble in water. Nonpolar and insoluble in water. Lipids are also composed of atoms of carbons, hydrogen, oxygen, but they do not have the 2:1 hydrogen-oxygen ratio. They are diverse in structure but share the property of being soluble in non-polar solvents such as ether, alcohol, but not in water. Examples are fats, which are formed from glycerol and fatty acids joined by an ester link. Saturated fats have no double bond. Unsaturated fats have several. Important in the membrane of cells are phospholipids, which also contain phosphorus, nitrogen, and sulfur. Lipids, called steroids, members of steroids, are important constituents of the plasma membrane in cells. Simple lipids, called fats or triglycerides, contain glycerol and fatty acids formed by dehydration synthesis. Unsaturated fats have one or more double bonds in the fatty acids. These are examples listed below: palmitic acid, stearic acid, oleic acid. Complex lipids contain hydrogen, oxygen, phosphorus, nitrogen, and sulfur. Membranes are made up of phospholipids. Saturated fatty acids and unsaturated fatty acids, noting the glycerol, phosphate, and organic groups. Steroids consist of 4 carbon rings with a hydroxyl group attached to one ring. These are part of the membrane. Proteins also essential in cell structure and function. Enzymes are proteins. Keep that in mind. Enzymes are proteins that speed up chemical reactions. We already talked about enzymes and how they facilitate the decrease in the activation energy in reactions. Transporter proteins move chemicals across the cell membrane. Flagella are made up of proteins. Flagella are structures, typically structures that help in the locomotion of a microorganism or a cell. Some bacterial toxins are proteins as well. Amino acids are the subunits of proteins, which are organic molecules containing carbon, hydrogen, oxygen, nitrogen, as well as some sulfur. Amino acids have at least one carboxyl group, one amino group, which is attached to the same carbon atom, the alpha carbon, hence the name alpha amino acid. Amino acids exist in configurations called stereoisomers, which are designated by D or L. Only L isomers are found in biological proteins. You need to be familiar with that. L isomers found in biological proteins, except for the relatively few found in some bacterial cell walls and some in antibiotics. Only 20 different kinds of amino acids occur naturally in proteins. Amino acids in proteins are connected by peptide bonds. Proteins have 4 levels of organization that you should be familiar with: primary, secondary, tertiary, and quaternary. The names are easy to remember. A description includes primary, the order in which the amino acids are linked, their sequence, basically. Secondary, localized, repetitious twisting or folding of the polypeptide chain that forms helixes and pleated sheets held together by hydrogen bonds. This is the twisting. Tertiary structure, 3-dimensional shapes in which the sulfhydryl groups can form covalent disulfide links, hydrogen bonds providing the holding force, and finally, quaternary structure, an aggregation of 2 or more individual polypeptide units that operate as a single functional unit. A protein that loses its characteristic shape has undergone what's referred to as denaturation. It's not functional any longer. Conjugated proteins are formed by the combination of amino acids and other organic or inorganic components. Glycoproteins, for example, contain sugars. Nucleoproteins contain nucleic acids, and lipoproteins contain lipids. The following table looks at the 20 amino acids found in proteins. You can read through the list. Important to be somewhat familiar with these, though I will not ask you to be able to restate all of the 20 amino acids found in proteins. But it's a good idea to be familiar with some of these. Some are a little more recurrent than others, and we'll go into detail about those as each individual amino acid comes up later in the lecture series. Amino acids consist in either stereoisomers, D or L. L form is the most often found in nature. You need to be familiar with that. Peptide bonds between amino acids are formed by dehydration synthesis. You need to be familiar with peptide bonds, where you would see those at most often. The primary structure is a polypeptide chain. The secondary structure occurs when amino acid chains fold or coil into regular helix or pleats. You can see the helix and you can see the pleats in this illustration. The tertiary structure occurs when the helix folds irregularly, forming a disulfide bond, hydrogen bonds, and ionic bonds between the amino acids in the chain. Here in the illustration, you can see clearly the disulfide bonds. The quaternary structure consists of two or more polypeptides. Two or more polypeptides in a folded state. This is the quaternary structure. Now, the levels of protein structure. Conjugated proteins consist of amino acids and other organic molecules, glycoproteins, nucleoproteins, and lipoproteins. They consist of nucleotides, or nucleic acids consist of nucleotides. Nucleotides consist of pentose group, a pentose sugar, a phosphate group, and a nitrogen-containing purine or pyrimidine base. You do need to be familiar with nucleotides and what their components are. There are two principal nucleic acids, and you should be familiar with these: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. The basic units of nucleic acids are nucleotides. Each nucleotide unit of DNA contains three parts: a nitrogen-containing base, and either adenine, guanine, cytosine, or thymine. You should be familiar with these nitrogen-containing bases. Adenine and guanine are double ring structures, purines. Thymine, cytosine are single ring structures, pyrimidines. Note in the figure how adenine always pairs with thymine and cytosine with guanine. You should be familiar with these pairings. The complementary pairing allows one strand to be reproduced from the structure of another. RNA differs from DNA in several ways. It has usually a single-stranded 5-carbon sugar and ribose, and uracil replaces thymine. Important to remember that the uracil replaces the thymine in the nucleic acids. Nucleosides have a purine or pyrimidine attached to the pentose sugar, but have no phosphate group. The principal energy-carrying molecule in all cells is adenosine triphosphate, or ATP. It consists of adenosine unit and adenine and ribose joined to 3 phosphate groups. It releases much energy when converted to adenosine diphosphate by the addition of water molecules, which separates the terminal phosphate group. ATP is generated by energy-yielding reaction in the cells, such as decomposition of glucose. The energy carried by ATP can be used to perform synthesis. This is sometimes referred to as the Krebs cycle, where glucose is broken down into energy. Each DNA has deoxyribose. It exists as a double helix, a hydrogen bond with a thiamine, adenine hydrogen bonds with thiamine, I guess I should say, and cytosine hydrogen bonds with guanine in the structure. You can see the bases that are apart: thiamine, adenine, cytosine, and guanine. RNA has a ribose, has a single-stranded adenine hydrogen bonds with a uracil, and cytosine hydrogen bonds with guanine. ATP has ribose, adenine, and 3 phosphate groups. It's made by dehydration synthesis and broken down by hydrolysis to liberate energy in the cell. Adenosine triphosphate plus H2O, adenosine triphosphate, water. Adenosine triphosphate plus energy yields adenosine diphosphate, or inorganic phosphate.